Everyone? Can everyone hear me?
Yeah, we can hear.
Okay, I guess it's almost 6:20, so we should probably go ahead and get started. I can introduce myself. Um, my name's Akshay Nagar. Well, actually, let me check to make sure everyone's in here. It looks like we might still be waiting for some people. Maybe I'll give it another minute, actually. It's until 6:20.
Hi, Professor. I will have to play the videos if you.
Okay, so it's 6:20. I guess I can go ahead and get started now. So yeah, my name is Akshay Nagar. I am a graduate student at Brown University, and I am also presenting at this conference, but in a different session. And I will be the chair for this session today. We have a number of presentations to get through, and I will be sticking to the allotted time, which is 15 minutes per presentation. So please know that whether you're presenting live or whether you're doing a video, once your presentation hits the 15-minute mark, I will be interrupting and for the sake of time, asking us to move on. So yeah, are there any questions before we get started? Okay. Yeah, and there will be time for questions afterwards if the speaker is in the session. If you're not going to be able to attend, or if you're not in the session, or if you would prefer your video to be played, please let me know, and I'll play it for you. Okay, so first up, we have Young Min Song from Gwangju Institute of Science and Technology in Korea. And they're going to be talking to us about passive radiative cooling structures for applications in outdoor usable patch type wearable devices. So if you could share your screen and unmute yourself, then start with your presentation. Are they in here? Maybe they're late. Young Min Song doesn't look like they're in here. And I don't think they sent me a video, actually.
I could swap presentations if it's helpful.
Sorry, what was that?
I could go first if it's helpful.
Sure. Why don't we do that? So instead we have Cody Schlenker from the University of Washington, who'll be talking to us about photon up conversion based on heavy atom-free thiosphoranes. Okay, so Cody, please go ahead. Thank you.
Well, first of all, thank you to the organizers for a kind invitation. Thank you to the host for moderating today. So today I want to tell you all a little bit about some of the work that we're doing in my lab at UW, focused on photon up conversion using dye molecules that are based on organic materials. Hopefully you can hear me okay. Just a little bit about things going on in my lab in general. We are a physical materials lab. We make stuff, we measure stuff. Usually those things are targeted towards energy applications such as photovoltaics or solar fuel production or electrochemical energy storage. What I'll talk to you today about is primarily focused on molecular photochemistry, but a little bit of physics. So as you may all be aware, one of the problems of solar energy conversion is that part of the light from the sun is in a region of the spectrum where most photovoltaic materials don't absorb well. Silicon does an okay job, gallium arsenide does an okay job, and organics do an okay job in the visible region of the spectrum. But out to these near infrared regions, it's kind of hard to absorb light and convert it to electricity efficiently, and there's a trade-off between energy and efficiency that you can produce because of the shock absorber. There have been some attempts to use this portion of the solar spectrum by various processes, taking low energy photons and converting them, and that's what I'll talk about today a little bit. One of the challenges, obviously, is you get to the lower energy region of the spectrum. The lifetime for materials, particularly organic materials, but all materials really, starts to get shorter and shorter and shorter based on energy gap law. And what I'll try to hopefully convey today is that if you can control some of these processes that go into energy gap law, you may be able to actually do better than one might assume. And that will be the target for what we're looking at here in the future. So what is upconversion and why do we care? Upconversion is essentially taking low energy photons. So for example, here's a photograph of a red laser beam shining onto a sample that has some upconversion special sauce in the cuvette here. Taking those low energy photons in the red region and converting them to high energy photons. So basically taking the energy of 2 photons and mixing them in some way. In this case, it happens to be in a photochemical process to generate higher energy photons. As you can see here, this could be helpful to various degrees in different types of solar energy applications. Here what I'm plotting is the energy gap of the solar cell, some type of photovoltaic device, versus the efficiency that you get out for a combination of multi-exciton generation or upconversion. And as you can tell, the differential of benefit that you get is sort of based on the gap of your cell. So for silicon, upconversion gets you something. For cadmium, maybe it gets you something a little bit more. And so you might look at the band gap of cadmium and say, okay, well, if you could get some dyes that are absorbing beyond, 825 or something, into the near infrared region, maybe you could be doing something that could be useful. So what type of upconversion might we be thinking about? Well, the type of upconversion that we look at is triplet annihilation upconversion. So you've got some molecules, they're banging around, they're absorbing light. They can maybe transfer or undergo intersystem crossing from a singlet excited state into a triplet excited state. If you can do that enough times, maybe you can get a bunch of triplets in solution that can start having the triplets interact with one another, and then they can undergo a process called triplet annihilation, which essentially is a spin conserved process that takes 2 triplets, fuses their energy into one singlet state in an overall spin conserved process that is more or less summarized here, where you have electron exchange between 2 triplet excited states, and it overall forms a spin singlet excited state and a spin singlet ground state. So you have this process of annihilating 2 triplets and generating one singlet. Okay, so the tricky part about all this is that you've got to have a sensitizer that can take light and turn it into take a singlet excited state from a photon absorption and turn it into a triplet excited state. You can do this with lots of different types of molecules. Here's a bunch of examples that primarily are precious metal compounds. There are a few porphyrin compounds and other materials in here that are not precious metals. But you really want something that's spectrally clean, where you can stick the absorption where you want it and it doesn't interfere with other stuff. You don't want to have to use precious metals. You also don't want to have to use things that are photolabile. So if you start sticking iodine in there as a sensitizer to induce intersystem crossing, you start getting all kinds of weird photochemistry going on, and these types of molecules bleach pretty quickly. All right, so what am I going to tell you about? Well, what we did was we took a dye absorbing molecule, which is a sulfur substituted chromophore here, which is a thiospray molecule, which essentially takes advantage of something called L-Syadrol, which I'll tell you about in a second, and utilizes the change in the orbital character in order to drive intersystem crossing with this molecule, and then you can be off to the races with your triplet annihilation. So what's L-Syadrol? So L-Syadrol basically says, hey, if I introduce some non-bonding orbitals below my bright state of my absorbing molecule here, I've now got this state, which is an end of pi star non-bonding orbital excitation that hangs around and just waits for something to happen. It doesn't emit very strongly. And if I've got a triplet level here that I can, or a triplet state that I can intersystem cross to, I can undergo spin orbit coupling based on the coupling of that end of pi star, the triplet pi to pi star state, essentially looking something like this. And so this orbital energy alignment of end of pi star and pi to pi star for singlets and triplets introduces some spin orbit coupling character, and that's L psi. So that's essentially what we've taken advantage of here. Okay, so we make some molecules, we make these squaring molecules. It's pretty much a quick reaction where you've got a condensation reaction of a squaric acid, and in our case, we've got an aniline donor unit, and then we thionate it with Lawson's reagent. And so you can see the absorption here. It shuts off the fluorescence when you thionate and introduces some process for spin over coupling. We use transient absorption to monitor the excited state kinetics and for these things, so we can watch the intersystem crossing process. Here we can monitor the change in the drop in the single absorption and introduction of the triplet of this TSQ molecule, and we can monitor the time scale for that. It's about 5 seconds. That's pretty good for having no platinum or iridium around. And so we can see the growth of the triplet there, so we can kind of monitor the kinetics for that. We can also monitor the kinetics for the triplet of our excited state, and we can see that lasts for about 20 microseconds. So this is the K of the triplet of our excited state here. We can then add in some emitter molecule or something to quench that triplet, and we'll use, in our case, just we'll use a pretty standard molecule, which is a rubrene molecule. It's a standard molecule for these types of upconversion experiments. It's not really that high performance, but it at least helps us prove the concept of what we're looking at. And so this molecule can quench the triplet energy of our dye molecule. And so here you see the native lifetime of our triplet for our dye molecule. We add rubrene and it quenches that. We can see the growth of the rubrene triplet as well. You pair these things together. So here is rubrene just by itself. We see no fluorescence from the rubrene whatsoever if we excite it in the red region. If we excite our dye by itself in the red region, we see no emission as well. Here is the emission from the rubrene if we excite it into its absorption. We see 450 luminescence. If we take the mixture of our two materials together, we now see what used to be dark in both cases. Red excitation of each of these individual components by themselves leads to no fluorescence. We mix them together and we now see the upconverted fluorescence from the rubrene. With no platinum, iridium, rhodium, rubidium, anything like that around. Okay, so this is essentially the process that we're looking at here. The nice thing about this is one of the attractive things for solar energy applications is that you can do this under broadband excitation. So this is a solar simulator here with a red filter. So we basically have a long pass filter here that just lets red light through from the solar excitation, and then here is the emission from our conversion material. You can also put these molecules into a film. So here is a film in our device chip where we're shining a red laser onto the film. We've got a color filter on the camera here that allows us to block out the laser excitation, that you can see the upconversion coming out of the film there. Okay, so I pitched this idea that we can take heavy atom-free materials, and they may have some potential for solar energy applications. And then I showed you some red excitation, which is clearly above band gap for a CAD telluride cell. What we've done now, and hopefully this paper will be coming out soon, but what we've done now is we've gone through and we've taken the photophysical parameters that we've collected from the kinetics measurements that we've got, and we've gone through it and we said, well, what does energy gap law say about the rate constants for the non-radiative decay? What does the Strickler-Berg relationship say for the radiative rate constants? What have we measured for the intersystem crossing times? And what are some reasonable predictions that we might come to for this class of molecules if we were going to do something, like try to harvest photons that would be sub-gap for a cell like cat telluride? And what was actually really surprising to us about this when we did all of this from the fundamental calculations of the kinetics is that actually for quite reasonable relaxation energies, which is basically 1/2 of the Stokes shift for these types of molecules, you can get to really quite respectable intersystem crossing yields for triplets, almost approaching unity. For physically attainable, for parameters that have been attained in the literature already for dyes, you could, pretty reasonably design a dye molecule that's going to undergo relatively efficient intersystem crossing. And so that's what we're doing now. And unfortunately, I can't tell you, or lawyers tell us at least that we can't tell you what any of those molecules are right now, but hopefully sometime soon we'll be able to talk in more detail about some of that work. And so the idea would be that we can take one of these new dyes that we're developing and actually start in real life up converting some photons that we could be kicking back to a device that could give us out some real benefit in some of these photovoltaic applications. So overall, I think that they're in a place where it would have been even 5 years ago, probably commercially not viable to even think about coating square kilometers with platinum or iridium complexes. I think that there may be some real potential for growth in photon up conversion for real solar energy applications here. I think that the squaring dyes offer some neat photophysical parameters to play with. I think there's a great test bed to play with there because of the modularity of their absorption and their spectral fingerprints. I know I'm probably over time here, but anyway, I think the future looks quite interesting for this class of materials. So stay tuned. We hope to have some neat results in this area. I want to say a big thanks to the students that have done this work. So Katie Corp started off this project. Sarah Pristash has now taken it over and is hard at work on some of the stuff. She'll be graduating here in the next year or so. Also thanks to the National Science Foundation and the Clean Energy Institute here at UW for support. And I will take any questions now, and I apologize for being a little bit over time.
That's okay. Thank you, Dr. Schlenker. Are there any questions?
Come on, I know Bruce has one.
I sure I could ask you a question. So can you tell me what you do? What's the stability of these guys? Is it they work best in films, or is it more you can do them in suspension? What do you?
It definitely depends on clearly when you have unity intersystem crossing yields for these things, you've got carbon, hydrogen bonds around, and if you have any oxygen around, you've got singlet oxygen right away. So as long as you can keep the oxygen out, they're pretty stable. And at least as far as we've seen, so we can, we've run up conversion experiments in solution in a totally anaerobic environment for days and days and days at a time. We don't see any degradation. There does appear to be some photothermal effects with some of our really high energy up conversion acceptors. So if you start pounding on the thing by heating it up, to 60, 70 degrees while shining light on it, there seems to be some sort of photothermal activity, but just light, room temperature, we don't seem to see anything. In films, it's much more stable. You can do up conversion in air in a film, and it doesn't seem to be a problem there. So yeah, they appear to be much more stable than any of the halogens out there.
Yeah. I'm just wondering if you have any like encapsulation technologies or things like that let's say for putting on a film to make it even, last even
Yeah, I
longer?
mean, what we've done in the past with just, very rudimentary is just, seal the thing in epoxy in a glove box and, they last for years that way, you know. But they're not out, being hit by birds and, having, dust thrown on them and people
Right,
walking on them and stuff, out in the field in real life. So yeah, that's, there's so much good encapsulation technology out there that we haven't worried too much about that. If we can, in our hands, if we can get enough oxygen out that we can measure it for weeks at a time without really, trying too hard, we feel like that's, somebody out there can encapsulate stuff better than we can.
Yeah. Okay, great. Thanks.
Yep. Thanks, Bruce, for the question.
Okay, thank you very much, Dr. Schlenker. If there are no more questions, we can move to our next one. So is there anyone, or rather, is Yahong Xie here from the Department of Material Science and Engineering at UCLA? Okay, I have their presentation, so I can share my screen, and I think it's this one. Oh wait, actually, excuse me. So let me. Yeah, I'll share my screen now. Okay. So can everyone see what's on my screen right now?
Yeah, we can see.
Okay, so this is Yahong Xie from UCLA.
Excuse me, Professor, we cannot hear the sound.
Sorry, what was that?
We cannot hear the sound.
Oh, okay.
I think when you are sharing, you can click the share sound.
Okay, okay. Let me do that. Sorry, everyone. Let me figure out how to do that. Okay, and then let's try this again. This is better?
Sorry, we still cannot hear.
Okay.
There's a setting in Zoom about your sound source.
Yeah,
hi, Professor. Maybe I can share the video.
Yeah, so, okay, yeah, go ahead.
Hello, my name is Yahong Xie. I'm with the Department of Material Science and Engineering at UCLA, and listed here are my collaborators. And the title of my talk today is Surface Plasmonic Sensors for Biomedical Applications, with an emphasis on light biomedical matter interaction. Here is the outline of my talk. I'll first give a brief introduction to the physics of SERS, surface enhanced Raman spectroscopy, and that combined with machine learning enables us as a research group to venture into the biological world where engineering meets biology. And there we encounter a clinically relevant question, which is disease diagnostic accuracy and the critical role of machine learning. I'll provide a number of examples throughout our study or research, and I'll wrap up with a future outlook. First, on Raman spectroscopy, I presume majority of you guys actually familiar with what is Raman process. at the core is a form of interaction between photons and phonons. The key advantage of such interaction is that phonons have only allowed wave number or energies. Therefore, by analyzing the spectrum of the scattered photons, we can backtrack into what the material we're looking at. Now, combined with surface plasmon resonance, a Raman signal can be amplified by orders of magnitude. Therefore, we call that surface enhanced. So it's SERS. And when SERS platform is used for analyze biological entities, in this case, I show a cell with a nucleus and a plasmonically active metal surface. When the beam of laser light shining on this surface plasma resonance is excited, and that actually in turn result in a very strong SERS scattering or Raman scattering signal to be collected and analyzed. Now, there are a number of outstanding characteristics of SERS, one of them being the single molecule sensitivity. And that is truly an amazing feature. We can get a fingerprint, spectral fingerprint from one single molecule. And that really gives the sensitivity to the technology. And how do we know is whether a spectrum is from a single molecule? This is classically done through the study of a bianalyte. And there you can get a definitive signature or conclusion that whatever the spectrum you get is whether it's from a single molecule or is from more than one molecule. Now, second part is the highly specific nature of SERS spectrum. Now, to validate that, my group conducted a study, examination of a kind of a peptide commonly present in human brain, and we call that amyloid beta 42. And this presence of amyloid beta 42 is associated with plaque formation in our mind, especially in the minds, in the brains of the Alzheimer patients. So there, my student conducted 196 hours nonstop scan of the spectral features as shown in this PCA plot, principal component analysis plot on the lower right. And there, with time, we can see each of the dots here, it represents one spectrum, or one SERS spectrum. With time, the SERS spectrum moves across the principal component phase space. And when we get to 196 hours, they pretty much actually after 24 hours, the spectral feature do not change much. And it's all clustered and become tighter and tighter, more tightly distributed. Okay, and also SERS is a technology that commonly perceived as non-quantitative, mainly because SERS is induced by surface plasma resonance, which is highly dependent on the detailed nanometer scale metallic structures, which we don't have that precision of a control. Now, to that end, my group invented a SERS platform that with a layer of graphene coated on there. And there, by looking at the graphene spectrum, the G peak of graphene is directly related to the local intensity of the plasma resonance. Therefore, the single layer graphene serves as a built-in plasma resonance intensity gauge. With that gauge, we can normalize all the molecular spectrum or signature relative to that gauge, thereby enable us to have quantitative SERS measurement across many orders of magnitude. In other words, with a large dynamic range. Now, another, so the after we get the search spectrum, the important thing is how do you analyze that? How do you tell its similarity or differences? There we have to employ machine learning. And the important message I want to say here is the picking and choosing of the specific machine learning algorithm is awfully important to the eventual accuracy of the biosensing result. And here we are using this particular combination of algorithm, starting with hierarchical clustering analysis and associated labeling in analyzing exosomes from patient samples. And the machine learning then go through a neural network-based a classifier for repeated cyclic cross-validation and training. And finally, it goes through a blind test. And this kind of learning process typically rendering a learning accuracy versus learning epochs or the cycle we run the machine learning through, and it typically shows this behavior like shown in the green curve. In other words, with the increasing number of epochs, the learning accuracy become higher and higher. However, upon cross-validation, as shown in the red curve, you can see that after a certain number of epochs, the actual accuracy no longer increase. And the distance between the green line and the red curve is known as oversampling. And that's something to be watched out for when you're doing machine learning. Okay, so when engineering and the technology platform like SERS meets biology, the first lesson, at least our group have learned, is how to handle variability. In engineering, we're not so used to a huge amount of variability. As such, if we repeat a physical science or engineering experiment, if you do it in a repeated fashion, the result will come out to be exactly the same. Whereas in biology, it's not exactly, because nothing in biology is exactly the same. Everything has a huge variability behind it. Therefore, in order to get meaningful result, we have to have a we have to be highly cognizant of the importance of statistics. Now, from an engineer perspective, I always have to come up with an explanation for myself as to why biological world is so highly variable, whereas engineering or physics world is highly repeatable. Now, I think this is my tentative explanation I exchange with this audience, which is the key point is the bonding strength. Like in biology, there is a kind of bonding called hydrogen bond. I remind you that hydrogen bond is not like a hydrogen atom and hydrogen atom bonding. It's a so-called hydrogen bond is a form of weak bonding, very similar to van der Waals bonding. And the bonding strength could be orders of magnitude lower than that of a typical covalent bond, such as silicon-silicon bond. Okay, in that case, then everything is, the barrier of one configuration or another, it's very low at room temperature, and therefore they change a lot. Now, you ask, what's the importance of hydrogen bond? Well, hydrogen bond is of overriding importance in biology because just to mention a few, DNA, the double helix, the two helix is bound together via hydrogen bond. And for RNA, as in the figure shown in the middle, all these black lines shows the hydrogen bond. Hydrogen bond is what holds the molecule in a shape. We call it conformation, right? For proteins, as shown by the illustration on the right, is the same. Hydrogen bond is what holds the shape, and the shape ultimately determine the biological functions. That's why hydrogen bond is important. And it's important, but have a very low making and breaking the energy barrier for that. Therefore, the biological world is highly, highly variable. Now, having recognized all those, we want to show that still this SIRS combined with machine learning, we call it SIM, a SIRS identification of molecules. This is our platform. SIRS combined with machine learning still can be very, very useful in disease diagnostics. And this is on this slide, we show the first example from our group's research, which is biomolecule based. And in that case, we analyzed 28 subjects of Alzheimer's disease, analyzed their cerebral spinal fluid. And we look at them and using SIRS, and we have achieved a 94% accuracy in cross-validation using convolutional neural network. And the diagnostic outcome is, we don't have time to go into details, but this table basically shows that the diagnostic outcome is highly correlated with cognitive test outcomes. In there, we then performed a double-blind test using 5 patient samples multiplied into 15 samples, and we cluster them into groups and into these 5 groups. And we then, after the unblinding, we found 100% match of these groups. And this slide shows a sort of a superimposed spectral feature within each of the groups. And you can see the similarity within each group and the dissimilarity or differences in between groups. So that's this one, it serves as the first feasibility demonstration of a biochemical-based liquid biopsy for Alzheimer's disease. Now, the next example we're going to show is our application of SIM to disease diagnostics through exosome-based analysis. Exosomes, as shown by this illustrative figure, are the part of individual cells' normal biogenesis. When cells live, they actually routinely secrete exosomes, which is a small particle on the order of 100 nanometer in size, but they carry important information. The biological field, people have increasingly recognized the value of exosomes. So in our case, we use SIM to analyze exosomes that we got from people's either separated from blood of patients or tissue or saliva. And the challenge there is when you get these exosomes, they are naturally secreted by both healthy cells and disease cells. The challenge for us is to identify the ones that secreted out by disease cells, and we're using their biological signature to diagnose disease. Now, first of all, how do we know what we're looking at is exosomes? Here, these 3 images represents the 2-dimensional scan of our substrate. And if we see particle like this with a very small spatial extent in which both the protein signature, the protein peaks and the lipid peaks and the nucleic acid peaks, they coincide in space, that's a good indication that—
Dr. Zhao, I think it's been 15 minutes, so we might have to cut it off here.
We use that to diagnose disease. We first, in this diagram on the left, each of the green dot.
Yeah, so unfortunately, it has been 15 minutes. That was a very interesting talk, if anyone in his group is here. Is there anyone from this presentations group here that is available to answer questions? Okay, if not, then we can move on to the next one. Next, we have from the Department of Material Science and Engineering at Seoul National University, we have Jae-young Bae. Are you here? Okay, I think I figured out how to share sound, so I can try once again. I think I have their Do I have their presentation? I actually don't.
Sorry. Yeah, I don't.
They don't have a video. Okay. All right, I suppose if they're not here, then we should move on to the next one after that, which is Bruce Cohen from Lawrence Berkeley National Laboratory, who will be presenting on photoavalanching nanoparticles. So Dr. Cohen.
Great. Let me share my screen. Hi, everyone. Thanks for being here. Let me figure out how to hide you all. Okay. Great. Okay, so I want to talk today a little bit about our work on developing new optical materials, particularly with relates to bioimaging. And this is something my group at the Molecular Foundry in Berkeley has been pursuing for a little while. And when we first started, we were working on materials like this movie. These are semiconductor quantum dots, so sort of more traditional nanoscience, if you can call nanoscience traditional, we're working with materials that are much improved versions of what particularly biologists had used for imaging for many years. But in a sense, the materials themselves were kind of the same. And in this case, this is a video of a single quantum dot, the red, and then there's a single GFP. These are on a protein that's walking along a microtubule. And the point you can see here is that the red persists while the green photobleaches. So what we have is essentially a material that is a much better version of what people have been using, but it's still kind of got the same optical properties. And lately what we've been doing, and this is now a tweet I'm showing you from our cover that Nature put out. But what we've been doing is we've been developing materials where you really can imagine sort of the proteins, the organics that people have been using in imaging as, being remotely related to this. These have far different properties. In this case, I'm going to talk to you today about photon avalanching. And this is something that we've isolated for a first time inside of a nanoparticle. And so the background here, and I'm just going to focus really on this one top point because it's such a short talk, that we can make nanocrystals that can do with light things that you simply cannot do with other sorts of materials. And I'll show you what I mean by that. So these sorts of materials are lanthanide based. There's up conversion. So we heard Kari talk about these a little earlier, this process of absorbing multiple low energy photons and then somehow summing that energy and emitting a higher energy photon. And in this case, we're working in the near infrared. So we're absorbing at 1064 and emitting at 800 nanometers. And I'll talk about a little bit why those wavelengths are so good for imaging living systems. And these materials are based on lanthanide. So these are these are F elements, not the radioactive types of elements that are made in synchrotrons, but naturally occurring elements. They're called rare earths. They're not particularly rare, although they can be tricky to mine. But we first started working with these a little over a decade ago, and this is a collaboration with Jim Shuck, my long-term collaborator who's now at Columbia, where we showed that you can image a single one of these under ambient conditions for over an hour, and they never photobleach. And we've repeated this many times on all the sorts of different ones we do. So right here and there, we have something where this is just something you really can't imagine doing with, say, green fluorescent protein or organic. And there were a few other things that we found. There's nothing in a cell that does this, that upconverts. Our cells don't have lanthanides. You don't have things like TTA inside of cells naturally. And so you can image essentially without background. And there were a few other things, these don't blink, that we discovered, but this really sort of set us in motion in trying to pursue these materials. And we had to work out a synthesis. So we worked out a synthesis. And one of the sort of joys of this material is that within this synthesis, you can dope into the nanocrystal pretty much any lanthanide at any amount. And this really sort of then set in motion a lot of our other work in trying to improve these materials so that you could really image them at very, very benign conditions. So we would do single particle characterization, varying the amount of lanthanide, say in the nanoparticle, all the way up to 100% doping. So we started calling these alloyed nanoparticles, and we showed that you could image these several millimeters into tissue. So this is in a live mouse using a 980. So this is near infrared light, so it's relatively low energy. About a fifth the laser intensity of a standard green laser pointer. And if we were in person, I would be making that joke holding my green laser pointer. But since this is Zoom, I just kind of drag my mouse over it. So anyway, these are exceptionally benign conditions. One of the things that really sort of drove us, particularly for these materials, is that near infrared light will penetrate tissue in ways that certainly ultraviolet, but even visible light will not. And this is simply a matter of scattering, which is somewhat large, well, in some cases, largely wavelength dependent. And also the fact that there are very few things in cells that absorb at these wavelengths. So this is a piece of mouse brain that is fixed. And my collaborator, Emery Chen, took an absorbance spectrum or transmission spectrum of this and showed that when you start to get into this near infrared 2, so between 1,000 and 1,400, you really have kind of a maximal transparency. And these lanthanides are the sorts of lanthanides that we are used interchangeably to dope into our nanocrystals. And so what we showed initially was you can take this combination. So this is a terbium and thulium. And the way our upconversion typically works is you have something that absorbs the light and passes it on through a kind of resonant energy kind of mechanism to a very neighboring lanthanide in the nanocrystal matrix. And that happens multiple times. It happens sequentially, and then that's how you get upconversion. And so here you can absorb at 980, which is where terbium absorbs very well, and get emission from thulium at 800. And you can see that through 2 millimeters of mouse brain. So this is sort of the nanoparticles are patterned on the surface. And you can actually get a spectrum of that image showing the 800 nanometer band. And what we spent a long time trying to do was trying to figure out wavelengths to use other than 980, both for doing multiplexed imaging, but also water has an absorbance in 980. And so if you get to a certain, confocal microscope power, you start to get water heating, which is not good. And a really classic wavelength is 1064. So there's neodymium lasers classically available at 1064, but there's this is from Arthur Ashkin's. This is possibly the image that won him a Nobel Prize in physics a couple of years ago, showing laser trapping of live cells. So these are yeast cells. This is 1987, so we have stills instead of a movie. But you can actually, these are cells that are trapped in the beam waste, so trapped in the laser trap, just dividing. So just going about their business as if they're not, stuck by a laser. And so this is just demonstration that this wavelength in particular is very benign for living systems. There's not a water absorbance, so there's not really heating, and there's really nothing in the cell that absorbs well at 1064. And what we did was, this was sort of an accident, but it actually turned into a successful story, is showed that if we excite these lanthanides at 1064, we're able to get this avalanching process. And so what is photon avalanching? This is, we are exciting these lanthanides first with one photon and then a second photon. And what this element thulium likes to do is, if there's another thulium close to it, transfer half of its energy essentially. So it's called cross relaxation. So you have a 2-photon absorbance event, and instead of emitting a photon, the kinetics of this are such that it will transfer that energy to a neighbor thulium much more quickly. And so what happens then is, now you start to excite these singly excited thuliums. This happens over and over and over again until you reach some threshold and you emit all the photons at once. That's photon avalanching. So the definition of avalanche is sort of like what you imagine like a snow avalanche on a mountain. You have a series of very small events that add up to something enormous. And so what that looks like is this. There's a series of events. Again, I just explained this, so maybe I won't go through it. One of the other things that's interesting here is thulium can absorb light as well at further into the near infrared at 1450 as well. But it doesn't actually have an absorbance at 1064. Thulium's absorbance peak is 980. So we are effectively exciting a couple of phonon vibrations away from that 980. So it's very counterintuitive that you can excite not at the peak and get much better behavior like this. And it becomes photon avalanching if the steepness of that nonlinearity, so this is a very nonlinear process, and in fact, it's 15. In other words, if the emission intensity is the 15th power of the excitation intensity as you change it along in that threshold. So that's the definition of an avalanche. We made nanoparticles, we did these experiments, and we were actually able to find cases where the emission intensity was up to about the 32nd power of the excitation intensity, while you are in this part of the S curve, the avalanching part of the S curve. And what that means is if you're in this part of the curve, if you turn your laser up 10%. So if you have kind of standard imaging, you turn your laser up 10% and you're in a linear part of that spectrum, you expect a 10% increase in emission. If it's a 2 photon event, you might expect around a 20% increase in emission. Here, for this sort of level of extreme nonlinearity, a 10% increase in our pump power gives us about a 2,000% fold, increase in emission. Okay, so one of the reasons this is interesting is if you think about the resolution possible in doing microscopy, there's been Nobel Prize on this. There's, quite a bit about super resolution. It comes back to this very simple equation that Ernst Abbe developed in the 1870s that essentially this is the full width half max, but say the resolution is a function of the wavelength and a little bit that has to do with the geometry of the optics. So this is a numerical aperture. And so in modern optics, NA is roughly 1.4 or 1.5. And so your resolution is the wavelength divided by 3. And then fast forward 125 years when Watt Webb developed multi-photon imaging for bioimaging anyway. Some theorists went back, looked at this equation for resolution and said, okay, well, if you have more than one photon, then your resolution is going to depend on this slope I've been showing you, the nonlinearity, so 1 over the square root of the slope. And normally, if you're doing 2 photon imaging, you're increasing your excitation wavelength by, say, twice of what it normally is, and you'd be dividing by the square root of 2, so 1.4. So you'd actually have worse resolution. But you see, what I'm getting at is now with our resolution, if you have a slope of 26, theoretically, for 1064 excitation, we should be getting a resolution, so at resolution, so not super resolution, of 70 nanometers. And in fact, that's what happens. So the top is calculations showing what this would look like if you have two of these nanoparticles 300 nanometers apart and you're changing the spectrum. Changing the excitation wavelength, and that's actually the experiment that we have. If you notice here, the slope goes down. It's not as steep either before or after this threshold event. So you would expect actually at higher powers, you would get worse resolution, and that's what we're seeing here. Actually, you can see this is the lowest power I'm showing you, and this is a slightly higher power, and the resolution actually is better at this lower resolution because we're at the threshold. And yes, so this is to remind me to say this is not super resolution. This is actually at resolution. We're not better than so, but you can actually do super resolution with these nanoparticles using the sorts of point spread functions, so Fiona type things, where you get a localization accuracy, where you count every photon and you sort of have a map and you get a geometric mean of where the photons are. And that gives us a localization accuracy of about 2 nanometers, which these are 20 nanometer nanoparticles. So that's somewhere in the middle of that nanoparticle. Okay, I think that's probably, maybe I'm over. I wanted to wrap up and thank, so I mentioned Jim, Shuck, and Emery Chan. These are this work as part of a long-term collaboration with the two of them, as well as for this last project, Youngduk Sa and Arthur, I'll just call him Arthur. And yes, funding and whatnot. I'd be happy to take any questions. Thank you.
Thanks very much, Dr. Cohen. Does anyone have any questions for him?
Yeah, can I just go ahead?
Return the favor.
Yeah. No, great talk. Really super cool stuff. I had a couple of questions, some about, I have no idea what the residence time of any of the nanoparticles is in the body, but I'm not a biologist, so I'm not going to worry about it. I wonder what's the time in between, the pulse width of your excitation and the time duration that it takes to build up to the avalanche point. Do you guys have a sense of how long that takes?
Yeah.
What's the timing that you can store excitation in the nanoparticle?
You hit on the exact point. So first of all, it's a CW, it's continuous wave we use, but there is a rise time to all of these. And it really is a build up of energy within the nanocrystal. It varies, obviously. It's less than a second, but it's not at a point where just doing simple scanning where we can, imagine doing live cell imaging, right? You would have to be doing fixed cell imaging. That said, there are kind of what we've shown and what others have shown is if you're using a scanning technique, you are seeding, so your laser beam is not uniform, right? If your laser beam is kind of half a micron wide, it's a Gaussian. And so it's much more intense at the center, and that's kind of the intensity. But there's quite a bit of photon flux sort of on the edges of the laser spot. And so if you're scanning over a sample, even if you're not exactly focused on a particular spot, you've started to kind of that rise time before you scan over it. So there are some sort of tricks like that where we can play, where we might be able to kind of seed all of it, the sample in 1064 light, but below threshold, and work to get faster rise times. But yeah, that's one of the reasons why you get that sort of resolution with scanning techniques, is because of the fact that the center of the laser beam is much more intense. And so you're able to sort of differentiate where the nanoparticles are with better resolution. But we do have to have sort of this buildup, as you mentioned. Okay. In terms of nanoparticles in the body, no idea. We're not putting these people. Yeah.
Is there a time I could ask just a quick, short question? Is that okay?
Yeah, that should be fine. Go ahead.
So this will show my ignorance about the physics of the system, but is the avalanche effect, does it require coherent excitation? Or could it, if you had high enough intensity, non-coherent light, would you still see it? I presume
I don't think,
but I don't know.
I don't think that, no, I don't think we require any particular kind of, so coherence or whatnot.
Because it'd be interesting to, my question of the lifetime and the coherence question leads to a third question, which I'm going to slip in really fast, is whether or not you could think about the timing of the system where you could store excitation to drive chemical reactions, that require multiple quanta.
Yeah, that's interesting. Right, anyway, if you could, no, that's an interesting idea, and that's not something we've thought of these for catalysis. So yeah, I haven't thought about that. The timing we've sort of thought of as a kind of a problem, right? Because especially if you wanted to do live cell imaging, you would, you would not get fast events. And so we're sort of trying to work on scanning techniques where we can speed that up. But the idea of storing energy, you've accumulated through light,
to dump it,
is an interesting idea.
Multi-proton, multi-electron reaction could be. I mean, we dream about wanting to try to do those sorts of things. So it seems very intriguing to me that you have a system that seems to do that. on the time scale, which could be interesting. Right?
Yeah, no, that is, and you know, these do emit thulium. I'm not showing this, but does emit in the UV and in the blue as well. Not as well, but it does. And so those might be the sorts of wavelengths you would want for doing chemical reactions rather than 800 nanometers, which is pretty benign. So great.
Cool stuff. Okay, thanks a lot. I'll shut up.
Thanks. No, thanks, Cody. Appreciate it.
Okay, thank you very much. Yeah, we have a little bit of extra time since people didn't show up. So if you guys want to get into longer discussions,
we can chat. Anybody else want to chat?
Yeah. Are there any other questions for Dr. Cohen before we move on?
Okay. Thanks, everyone.
Okay, thank you. So next up is, and I don't think they're here, so I will go ahead and share the video now that I've learned how to share with sound. So let me just pull it up. So this is molecular band terahertz detection and imaging structures from, and I'm scared I might butcher the pronunciation of their name, but it's from.
Hello, I am Livio Popasimil, and I will present the molecular band terahertz detection and imaging structures.
Can everyone hear this?
Applications in many parts of life, from molecular visualization
Yeah,
to
we can hear.
virology and technical application as safety, security, and science research. I am an executive director at Los Alamos Academy of Sciences and the president of EPMM-2 in Los Alamos. I'm a nuclear engineer and physicist and research scientist, initially for Los Alamos National Laboratory and now for these private companies. For more information on me, please feel free to look on website and do a search on internet. Well, from Star Trek, we had Internet of Things versus big data integration and very futuristic views of a device called recorder, which applied in a sick bay can make a non-invasive detection of almost all the sicknesses and anomalies in the.
Okay. Is there anyone else who can't hear clearly? I just got a message that it's maybe hard to hear or they're not talking very slowly. Is that a problem for anyone else? No? My volume is at max right now. Unfortunately, I can see maybe on this, yeah, maybe it's a problem with my computer. If Dr. Zhao, maybe if you could share the video on your screen, maybe it'll be better for everyone to hear.
Okay, I can share.
Thank you. Sorry about that, everyone. Technical glitches.
Hello, I am Livio Popasimil, and I will present the molecular band terahertz detection and imaging structures with applications in many parts of life, from molecular visualization to biochemistry, chemistry, virology, and technical application as safety, security, and science research. I am an executive director at Los Alamos Academy of Sciences and the president of EPMM-2 in Los Alamos. I'm a nuclear engineer and physicist and research scientist, initially for Los Alamos National Laboratory and now for these private companies. For more information on me, please feel free to look on website and do a search on internet. Well, from Star Trek, we had Internet of Things versus big data integration and very futuristic views of a device called recorder, which applied in a sick bay can make a non-invasive detection of almost all the sicknesses and anomalies in the body. Well, in the movie was also augmented by paranormal skills of some characters there, but I'm talking only about this advanced technology and may be possible in the future to make direct data to brain transfer in order to increase computer to human communication, making a kind of cyborg. Well, terahertz is a part of electromagnetic spectrum, and is not well covered today by the actual technology, because the devices that are working, microwaves, have hard time switching and working in terahertz domain, while the devices working in visible and near infrared has also hard time detecting terahertz, because it is at a very low energy photons in milli-electron volt, and is out of both, it's in the middle of the both domains of electronics and optics, and this device comes to mitigate this gap. From the various point of view, you have Raman spectroscopy, terahertz detection, and far infrared or Fourier transform infrared, and molecules like felodipine have a complex spectrum in infrared and have various contributions of various oscillation eigenmodes inside the molecules, and are depending on temperature, the molecular bond strength, and the proximity to other molecules and the interface. Basically, the movements are internal, rotation, translation, and vibrations, symmetric molecular displacement. There are many cases of molecules, and there are eigenmodes of vibration, like this is water molecule vibration and has about 3 eigenmodes, and these are manifested in molecular spectrum, as well as a difference for carbon dioxide that has a linear structure instead of an angle structure as in water, and that has different eigenmodes that will have different vibration bands. When you look at more complex structures as uranium oxide and in reaction with nitrogen and water, we see various vibration modes that are contributions from various nitrogen combination, carbon dioxide, and also uranium oxide contributions that gives you fundamental frequency in the lower and upper band, but you need a high resolution detection in order to decide what really is and what is the contribution coming from. Well, in a more larger view, you have the atmospheric transmission bands and attenuation. So where attenuation is minimum in electromagnetic waves from a few megahertz to a few gigahertz, you obtain a good transmission through the atmosphere. Then the absorption bands of oxygen, water, carbon dioxide appears, and the transmission is reduced, so you got a lot of absorption. And this is Planck spectrum that depends on the temperature, and so you have many applications in terahertz domain. I will not enter all because, but some applications are in low terahertz, 1 to 3 terahertz for transmission through the closing and security imaging, or could be also medical imaging. And you may have a multispectral comparison of various bands, depends on how many bands and of detection you have and how narrow they are. You have a remote sensor like a multispectral thermal imager that is good for application. In our case, we use electromagnetic waves and a resonant input structure that is 3 times larger than the wavelength and has a parametric amplifier that basically changes the resistors and amplifies current from space impedance of 377 ohm to the field effect transistor gate impedance of few mega ohms, and the voltage is increased by effect of charge polaron amplification. Another device detects the anomaly, so the terahertz voltage is not able to switch actual electronic device, but is able to perturb it, and this perturbation is detected and is further differentiated in delay time digital analog converter that has basically no dead time and may go up to few gigasamples per second, as large as electronics is reliable to do that. And this is a schematic drawing of the detection module that detects a single frequency and digitize it, fulfilling memory bank with fast direct access and even mode storage. For details on how the parametric amplification is happening is based on electric field behavior and creating polarons, charges that are polarized due to electric field, and implicit due to shape, spherical shape, is giving electric field amplification, while the impedance is growing. You have about energy or power conservation until it reaches the gate of a field effect transistor, and in a nonlinear amplification characteristic, it gives distortion to the signal if you pass a reference signal and you disturb with the terahertz, you'll got a disturbed signal that is different from the initial signal, and that is what electronics can detect, and this is how the amplification takes place, subtracting the difference and then amplifying it and applying to further stages. In another view, this is a resonant element that is in contact with air or vacuum and has input impedance of 377 ohms, and each stage is giving an increase in impedance, and that means an increase in voltage until it gets a high voltage able to disturb low current or single electron field effect transistor, where the difference is detected and amplified and is transmitted to an analog digital converter. Everything is smaller than an actual discrete transistor on LED, and being an electromagnetic resonance structure, it can be placed in polarized mode, where you have a longitudinal or transversal polarization of the wave, or circular polarization using other kind of structures. And you may agglomerate octave-based resonators into triangle shapes that will be the elementary cell of a multi-frequency array detector. And finally, you can group them, creating a kind of fly-eye imager, where it detects everything that happens in angular visualization. From the electronic point of view, after you detect the perturbation, you enter into an analog to digital converter that is based on a level sensor that recreates the maximum detected level, and leaves out a difference between the integer detection versus a signal that is extracted and amplified and put in various stages. And this is how the wavefront looks like. So after you extract that, you give a delay time into an LRC line that reduces the propagation speed, giving time electronics to become stable and getting out of the transitory modes. And that will create a number of bits detected at each stage, and you may apply up to 3 stages with good resolution, giving up to 1820 bit. To each bit, you may apply correction, CRC code or hammer for error detection and regeneration. And the competition is analog devices AD9625 that is able to have a few gigasamples per second and good range and bandwidth. It's a high-performance device that it does about the same as our system is doing. You may get applications in clinical imagery, in LIFs, in bio, in cancer detection, in detection of damages in the tooth and molecular modifications inside the tooth, using both transparency and resonant absorption. And here is how it looks in a leaf, the spectrum before and after watering, and the leaf magnification image. Well, maybe sometimes this technology might work as anticipated, or do even more. Now this project is in technology readiness level 3, and we look forward for more development in order to obtain an advanced technology and rely less on paranormal skills or intuition in medical application, biomedical, and research. Thank you so much for your attention.
Okay, thank you very much, Dr. Zhao, for presenting that on your screen. So I don't think there's anyone else from that group to ask questions to, so we'll move on. Next on my list is Better Artificial Photosynthesis by Ion Segregation Oxide Nanoparticles from Douglas Gueva. Try one more time to share with my screen, and hopefully people will be able to hear this one better. Just pull it up. Okay. So, beginning here.
Hi, my name is Douglas Gouveia.
Is this clear to everyone?
I'm a professor at the University of São Paulo in Brazil.
That is good.
We have been working for over 20 years in understanding, quantifying the phenomenon of segregation in ceramic oxide interface. And today, we'll try to show you how this segregation is very important for a better artificial photosynthesis. The term artificial photosynthesis is commonly used to refer to any scheme for capturing and storing the energy from sunlight and the chemical bonds of fuel. Heterogeneous photocatalysis depends on the surface and bulk, and two characteristics of bulk is most important: band gap and the electron hole recombination time. And the band gap depends on the light, energy of light, and determines the chemical energy for all chemical reactions, and the valence band and the conductive band, and all defects, and then soluble impurities. You change the level of defects inside the band gap. The recombination time is a function of the free pathway, and then it's very interesting because we have a direct relationship with this recombination time with the conductivity of grain boundaries in the nano oxide. Simultaneously to the bulk properties, the surface properties are very important for the heterogeneous catalysis. And the surface determines the number of adsorption sites. This number of adsorption sites depends on the crystallographic surface and the kind of surface you have, and the kinetic and thermodynamic of adsorption, because all reaction depends on adsorption of species in the surface. To increase the photocatalysis, we need then a lot of surfaces, and then we work with nano-oxides. These nano-oxides have nanostructure. This nanostructure have two main parts: the grain boundaries and surfaces. Grain boundaries are all interface between solids, and surface are the interface between a solid and a gas, or a solid and a liquid. The interface segregation is a spontaneous thermodynamic process for both grain boundaries and surfaces, and it's a very important phenomenon that change all surface or interface properties, and the stability of particles, the reactivity of surfaces and interfaces, the electrical conductivity. But what is interface segregation in a solid? Segregation, adsorption, and surface energy are strictly related to the surface axis. The surface axis is done by the number of moles by meter square, or by surface. And then when you have a surface axis and a rate of segregation or adsorption, the system reacts with a decrease of surface energy and then stabilization. Then the surface energy of pure material is decreased by this amount of atoms in the surface by segregation or adsorption, and the rate of adsorption or segregation. Then the gas, we have then the pressure of gas, and the next to the surface we have adsorption layer, and this adsorption layer determine the surface excess, and then you have a decrease of surface energy. It's the same case for segregation. When we have a solid solution, in the bulk we have the solubility, but next to the surface we have an increase of concentration. This increase of concentration is the surface axis. Some years ago, we proposed a method to know how is the surface excess by a lixiviation, selective lixiviation method. Then, for nanoparticles, the nanoparticles, the grain boundary and surfaces, we have an additive segregate on the surface and the grain boundary, and some additive soluble in the bulk. Okay. Then we have an insoluble oxide matrix, and the additive is soluble in acid solution. Then by lixiviation in nitric acid, for example, it's possible to separate the atoms on the surface, segregate on the surface from the atoms in the grain boundary and soluble in the bulk. Okay, then this part is insoluble, this part is acid soluble. Then by centrifugation, we have a separation of phase. Here it's possible to know how is the concentration of atoms in the solution by ECP, for example. And here it's possible to know how the concentration of additive in the nanoparticles in the soluble part. By this method, it's very easy to know how is the concentration of additive in the surface and the surface axis. Looking the thermodynamic behind the surface axis, we have, when you have free energy of segregation, the concentration in the bulk and the surface are not the same. And the free energy of segregation is approximately the same of the rate of segregation. The rate of segregation has 4 components. The first one is the difference of surface energy between the solid and the solvent. The second one is related to the solubility. The third is the difference between the atomic size or ionic size. Then when you have a big difference between radius, is more easy to segregate. And the last one is the electric charge related to the charge of ions in the system, then related to the depletion layer. One of the main consequences of surface excess is the stabilization of particle size. Then when you take this law of particle growth, then the particle growth for time t, the growth depends on the surface energy and diffusion. And we have some kind of curve like that, and the n here is 2 or 3. And when you change the surface energy or the diffusion, when you decrease the surface energy or the diffusion, we have a decrease in the final particle size here. And we see this because when you have a surface excess, we have a decrease of surface energy and then the stabilization of particle size. And then as a consequence, a direct consequence is the increase of a specific surface area for a long time of calcination. Now I want to show you some results about our work in the photocatalysis and try to connect the surface segregation with the photosynthesis and how this photosynthesis could be better with ion segregation. Then we have 2 works. One is the segregation of chlorines in zinc oxide grain boundaries, then the change in the conductivity and the increase of recombination time, the consequences in the photocatalysis. And the second group of results is the transition metals, doped titanium oxide, then the segregation and photocatalysis. In this slide, you can see the chloride segregation results on zinc nanoxide surface. These results were recently published in the Ceramic International, and I invite you to read this paper, and showed that there was an intense chloride segregation on the surface, but at the same time there was segregation at the grain boundary. The segregation grain boundary causes a destruction of an electrical potential barrier. It is well known in the manufacture of zinc oxide varistors, and this destruction of potential barrier at the grain boundary causes an increase in the conductivity. The increase of total conductivity, then the conductivity of grain boundaries and bulk, increase the free pathway for electrons and rows, and then the time of recombination increase, and then you have a more time for photocatalytic reactions. In this impedance spectroscopy diagram here, we can see the increase of chlorine in the zinc oxide decrease the total resistivity. Then you can see here the total resistivity decrease with chlorine, and consequently we have an increase of conductivity. And this semicircle here is referred to the grain boundary resistance, and then you have a stronger decrease in the grain boundary resistivity, and this has as a consequence an increase of the total conductivity. This is very important for have more time of electrons and hole without any recombination, and then increase the photocatalysis. We have chosen the photodecomposition reaction of acetaminophen to understand the effect of chlorine segregation on ZNO. After 108 minutes of UV radiation, we have here the specific conversion, and for two conditions. The first condition is the as prepared powder, and the second condition is for powders lixiviated with water. We know that chlorine ions Poisson photocatalysis like ZNO. Therefore, when they are present on the surface, the reaction is reduced. However, after lixiviation, the reactivity increases very much, and mainly at the highest concentrations. And you see, for pure zinc oxide, we don't have any difference before and after lixiviation, but for high concentration of chlorides, we have a big difference. It's very interesting because in this case, most of chloride after lixiviation is in the grain boundaries. Then we have a very big conductivity, and the photocatalysis of acetaminophen is very important when you compare with pure ZNO. And then this shows us that for chloride, it is important to have chloride in the grain boundary to increase the conductivity, but it's not so good to have the chloride in the surface because the Poisson the reaction of photocatalysis. And for fluorides, the effect is very, very important. You can see here powders with a very low concentration of fluoride. We have a very important increase in the photocatalytic activity after lixiviation. And then the fluoride and chloride on the surface is not very so good, but is necessary in grain boundaries. We have some results, and this time is titanium oxide containing some oxides like manganese, iron, chromium, and copper oxide. Then this metal oxide, titanium oxide, for all 1% mole prepared by Pechini method at 350 degrees, 15 hours in air. And then we see the same phase, is just anatase, okay? And the crystallite size decrease with doping for all samples. The specific surface area increase with doping. Then we can think about segregation, and the surface excess prove this. Then we have a surface segregation for chromium, copper, iron, and manganese in different proportion. And the acetaminophen decomposition, you can see here the square, red square is the titanium oxide, the pure titanium oxide. And with iron, we have the same kinetic of decomposition. For copper and manganese, the kinetic is lower than the titanium oxide, but for chromium oxide, you have really an increase of decomposition of acetaminophen decomposition. Then this is very interesting. Then we think that this transition metal have the property of change the valency in the surface and transfer charge for the acetaminophen and have the oxidation of acetaminophen. But the more interesting is that when you lixiviate the powders, we have the same comportment of titanium oxide. Then after lixiviation, we can see all system independently of the composition, we have the same compartment. Then you can see here with segregation here for lixiviate. And then all catalysis activity are related to the segregation. This is really a result that give us this possibility to understand the segregation as a very important factor for photocatalysis. In summary, the segregation at interface controls the particle growth for nanoparticles and at the same time increase the photocatalysis efficiency by increase the conductivity of grain boundary, increase the recombination time, and for segregation of transition metals, facilitating the transfer of charge, surface charge for the reaction. I want to thank the organizing committee to invite me to talk to you, and for our sponsors, RCGI, FAPESP, ANP, and Shell Brazil. Thank you.
Okay, thank you very much. He said he'd connect by cell phone, but it doesn't look like he's here. Anyways, so let's move on to the next one. Now we're going to hear from Smruti Ranjan Sahu. Are you in the chat? If not, Dr. Zhao, do you mind sharing your screen again so that we can hear their video? Because I believe they have a submitted video. Thank you very much.
I would like to thank the organizers for giving me an opportunity to present my work here in this conference, which is titled as the development of a 3D platform for the detection of bilirubin concentration present in human blood by employing surface enhanced Raman scattering and plasma enhanced fluorescence detection methodologies. Bilirubin is an endogenous pathological substance present in human blood, which is a major metabolite of hemoglobin. It gets deposited in the human bloodstream and not secreted out. And the presence of excess bilirubin in human blood can lead to a series of diseases which can be liver related and can also create neurological disorder, which can create brain damage and death even. And considering the advantages of SARS, namely, which can be a label-free analytical technique, which gives intrinsic unique molecular fingerprint information from various substances, and its high sensitivity, simplicity, flexibility, and fast analysis speed enables it to be an on-site point of care testing technological tool. And the laser source used in SARS or Raman spectroscopy can be manipulated to avoid photobleaching and photodegradation in sensitive samples, which can therefore enable to achieve long-term monitoring. And the unique ability of SARS to detect multiple components simultaneously present in various organic, inorganic, or biosamples can therefore help us to get insights into the unique molecular fingerprint informations. The experimental challenges involve the generation and utilization of 3D surface plasmons and the enrichment in 3D plasmonic hotspots. And as we are interested in both source and PEF analysis from human blood, so considering the facile and economic viewpoints, we needed to construct a single platform for both of these purposes. And finally, to detect blood bilirubin. So these are some of the widely used nanostructures, particularly to manipulate surface plasmons. Normally, we use noble metal nanoparticles. But the fact that the noble metal nanoparticles have copious amount of free electrons, and these free electrons respond coherently to any sort of disturbance from the external electromagnetic field. And this sets the electron cloud into acceleration. And the electromagnetic field enhancement produced such as can be utilized for various purposes. And the surface plasmon resonance is largely geometry dependent. For example, in the case of nanorods, a very little change in the aspect ratio of the nanorods lead to a shift in the plasmon resonance. So we are particularly interested in silver nanowires. As we can see here, these are two of the many configurations that we are particularly interested in. And in this configuration, the nanoparticles are intercepted by two nanowires. And here the case is opposite. The nanowire is sandwiched between two nanospheres. In fact, these nanospheres represent as intersection points or junction points in our analysis. So to see the electromagnetic field enhancement, we find that the hotspots are particularly generated when the nanowires are intercepted by the nanospheres. And the similar case is here in this configuration, when the nanowires are intercepted by nanospheres. So we see there is a generation of hotspots. So it indicates that the structural arrangement of silver nanowires can lead to more number of hotspots. So our next target was, what if we construct a wood pile nanostructure? So the construction of a 3D random crushed wood pile nanostructure can lead to the generation of more number of hotspots, which can be used to detect an analyte. So to construct the 3D random crushed wood pile nanostructures from silver nanowires, we synthesize silver nanowires by the well-known polyol method. And to construct the 3D nano platform, we first sprinkled certain amount of silver nanowires onto a glass platform and subject it to vacuum dry. And we repeated this procedure subsequently so that any sort of alignment would be destroyed so as to create an wood pile structured silver nanowires. So this represents the structural disarrangement of silver nanowires. And as the substrate is ready, so the analytes can be drop cast onto the substrate and followed by vacuum dry, which can then be used as a source or PEF analytical platform. So to evaluate the efficiency of our source substrate created as such from the 3D nano platform, we used a standard molecule R6Z. So here is the source spectra obtained from the R6Z by variation in nanowire amounts. And we can see optimized source response can be achieved by variation in the silver nanowire amount. And to acquire source spectra, we normally took 7 different measurements and then averaged. And to test its stability, we also tested the stability of the platform after 80 days. And to know its limit of detection, we varied the silver, the rhodamine 6G molar concentrations. And as you can see, it can detect rhodamine 6G even up to femtomolar level. So this proves our as constructed 3D nano platform to be an effective search platform. So to know the efficiency of our platform for plasma enhanced fluorescence imaging, so we can see here there is a great contrast between R6G and R6G mixed with silver nanowires. Pure R6G doesn't show any kind of fluorescence, but when it is deposited on 3D random crossed wire wood pile silver nanostructures, we can see an increase in the fluorescence. So using this fluorescence detection mechanisms, so it can be, so rhodamine 6G can also be detected in a nanolight. So in these 2 experiments, we proved that a single platform, which is effective for both source and PEF measurements. Then we went on to detect bilirubin directly. So to get a molecular fingerprint from bilirubin, we varied the concentration of bilirubin and analyzed the nature of the spectra. So this is the source bilirubin molecular fingerprint. And then we spiked bilirubin with fetal bovine serum. And also we observed the similar nature of the bilirubin molecular fingerprints in the source spectra. Then we went on to analyze bilirubin concentration in blood samples. These blood samples were provided to us by nearby hospital authorities. And these were collected from different patients with different concentration of bilirubin present in their blood, which ranges from 2 micromole to almost 360 micromole. And we used our as constructed 3D nano platform to detect bilirubin, to detect the concentration of bilirubin present in these blood samples. So as we can see, it also indicates that the molecular fingerprint of bilirubin is also present here. And to further analyze, we particularly focused on 2 source bands, namely 1245 centimeter inverse and 1600 centimeter inverse really. And as you can see, the trends in both the cases are similar with increase in serum bilirubin concentration. So this proves that our nano platform can efficiently detect bilirubin concentration in human blood. This is the plasma enhanced fluorescence from bilirubin. As you can see, silver nanowires do not display any fluorescence, and so is the case in bilirubin. But when bilirubin is deposited on our nano platform, which contains 3-dimensional random crossed wood pile silver nanowires, we can see the fluorescence behavior. So this is a fast detection method to know the bilirubin concentration present in the analyte. So again, we used the single platform to study both source and PEF. So to conclude my talk, we have utilized the 3D surface plasmons present in a specially designed 3-dimensional random crushed wood pile silver wire nanostructures to detect blood bilirubin concentration. And We also constructed a single platform for both source and PEF analysis. And in our case, sample preparation was very simple. No pre-preparation was involved or any other further modification. We demonstrated our technique to be label-free with high sensitivity and fast detection. And our 3D nano platforms can be deposited on flexible substrates so as to widen their usage in other applications. And also, it can also improve the detection methodologies in hospitals for the use of like portable Raman spectrometers to detect the concentration of blood bilirubin present in patients as an on-site detection platform. This concludes my talk, and I thank all the participants who are listening to my talk. Thank you again.
Okay, thank you very much. Next, we have from Strasbourg, we have Anna Bonfiglio. Are you in here at all? Nope. Okay, so then we should play her video as well. I think we have it. This is well, I don't want to butcher the name of the presentation, so you'll just see it here.
Good afternoon, everyone. First of all, I would like to thank the organizer for inviting me and giving me the opportunity to share with you today some of our recent work in the field of light emitting devices. In particular, I would like to talk today about some heterobimetallic complexes for application in light emitting electrochemical cells. So just a brief introduction for those of you that are not really familiar with the topic. Actually, so phosphorescent transition metal complexes, they are very interesting because they possess tunable emission color over the entire visible spectrum and sometimes even beyond. They have high photoluminescence quantum yield, and so it depends on the color. They have relatively high photoluminescence quantum yields, and they also possess long-lived excited state, which arise from the formally triplet nature of the excited state. So they found very interesting application in optoelectronic devices such as organic light-emitting diodes and light-emitting electrochemical cells. And in particular, iridium-based emitters, they found currently application in market technology. So what is the challenge? Even though they are market products, actually there is still some difficult challenges that have to be problems that have to be tackled. In particular, it's very difficult to make efficient and stable near and red and near infrared emitters as well as blue emitters. So in particular, so this means that basically the two extreme of the visible regions are very difficult to address. So what is the origin of the nature? What is the nature of the emitting excited state, which then will help us to understand how to provide some guidelines on optimize the excited state and the emission properties. So in those compounds, the emission of the excited state can be defined as a linear combination of three different states. So it's a combination of mainly triplet ligand-centered excited state, which actually mix by a configuration interaction with a triplet metal ligand charge transfer excited state, and also by means of spin-orbit coupling with a singlet metal ligand charge transfer state. So the radiative rate constant, which also is linked to the photoluminescence quantum yield, is also directly proportional to the third power of the emission energy, along with spin orbit coupling, matrix elements, and the oscillator strength of the compound. So the problem is that the emission energy is intrinsically small for red and near infrared emitters, which means which brings to intrinsically small radiative rate constant. On the other hand, there is also a second parameter that has to be taken into account, which is derived by the energy gap law, which is the non-radiative rate constant. In particular, the logarithm of the non-radiative rate constant actually scales with the negative of the difference between the triplet excited state and the ground state, which means that the non-radiative rate constant is typically high for red and near-infrared emitters. So if you now look at the literature, main efforts have been devoted to development of mononuclear iridium complexes. So they are currently leading emitters, but actually the presence of a second metal may bring some interesting properties. In particular, they will help us to better modulate the redox and the optical properties, to increase the structural rigidity, which means that now we can somehow counterbalance the energy gap law by decreasing the radiative rate constant, and we could enhance the spin orbit coupling. So the first example of bimetallic complexes were reported by a group of Balzani in 1992, and now the topic a few years ago started to become much more investigated in the literature. In particular, thanks to the examples reported by the group of Bryce and Williams. On the other hand, heterobimetallic complexes, in particular those containing iridium complexes, iridium metal centers, they are much less explored, is because the few examples known in literature, there are those that I show here in the slide, and this is because they require site-selective reactions in order to improve the the reaction yield, and also careful design of the ligand and the metal, the ligand environment and the metal, in order to avoid metal-metal electronic communication. So we're then wondering whether it's possible to enhance the emission properties in red-emitting heterobimetallic complexes. So we then came across an interesting ligand. Is a Janus type ligand which possesses two different coordination sites. On one hand, the acetylacetonate site, which is very useful for the preparation of luminescent complexes, and on the other hand, we have an heterocyclic carbene site, which is also very interesting because it's able to form very strong metal-ligand bonds. So we then use these ligands in order to prepare some linear copper and gold complexes, bis-carbene complexes, which now we use in a subsequent step for the preparation of heterobimetallic complexes in a rather straightforward manner by also using site-selective steps. So we can then, we were able to straightforwardly prepare a series of heterobimetallic complexes, and here, just for sake of time, I will present just two of those, in particular compound number 4 and 5, and we can compare those with the monometallic counterpart, compound number 3. So if you look now briefly at the photophysical properties of those compounds, we can see that along the series 3, 4, 5, we have very similar absorption spectrum. In particular, for 4 and 5, the absorption spectrum is basically superposable. We have just a tiny, very small hypsochromic shift for the singlet metal to ligand charge transfer band for the compound number 3. So thanks to the DDFT calculations, we were also, by including spin orbit effects, we are also able to associate those bands, that band to the singlet metal to ligand charge transfer involving the sacrometallic ligand. On the other hand, in the emission spectrum, we also observe very similar emission profiles attributed to the triplet metal to ligand charge transfer state, with a very small hypsochromic shift again for the monometallic complex. If you now look at the more into the detail, the photophysical properties of compound 3, 4, and 5, and we compare that with the benchmark complex, the iridium bispicolinate ACAC that I have here in gray. So we can see that in particular, compound number 3 versus the benchmark complex, we have very similar photophysical properties. This means very similar photoluminescence quantum yield and very similar radiative rate constant. On the other hand, the benchmark versus the heterobimetallic one show very different, very different properties, and will help us to better understand to gain more details into the excited state properties of those heterobimetallic complexes. In particular, when we go from the monometallic to the heterobimetallic ones, we have almost twofold increase of the photoluminescence properties. We have very similar excited state lifetime, and more interesting, we have so the results is that we have almost twofold increase of the radiative rate constant for the heterobimetallic ones. So if you now
sorry for everyone, because the time is limited, I will just jump directly into the summary.
Near infrared region and here I show just briefly one of our latest results. So this actually brings me to the last slide of my talk of today, in particular the most important slide actually, which are the people that actually
sorry, we'll stop here, and all the videos are available online. Sorry.
Thank you, Dr. Zhao. So next we have, let's see, I don't know how to pronounce his name, Stin Yukin from KU Leuven in Belgium. Are you here? Anybody from that group here for, what is it, quantum dots for 2-photon cell transmembrane electric field imaging? Okay. Then next on the list is Mehardaj from the Indian Institute of Technology in Madras. Are you here? Sorry, could you repeat that?
Presentation.
You'll have to repeat that one more time. Your audio is a little choppy.
Can I share the presentation?
Sure. Yes.
Yeah.
Yes. Would you like us to show the video or do you want to present live?
I present it live. No issue. I will present it. Hello. Can you hear me?
We can hear you a little bit, but your audio is very choppy. It's cutting out a lot. So
No.
maybe it's better if we play your video.
Okay. Yeah. Can go ahead. Yeah. No issues. Yeah.
Okay. So Dr. Zhao, could you pull up her video, please?
Hello all, I'm Mehertaj H from Indian Institute of Technology Madras, India, and I am here to present for the conference AAA-FM UCLA 2021 on the topic the modeling of magnetogeological gels, a size effect study. So the topic I am presenting on now is the modeling of magnetogeological gels, a study on the particle size effect. So the outline of my presentation, will be starting off with the introduction, followed by the mathematical preliminaries involved in development of the model, followed by the model development statement and the total stress derivation, surface energy concepts involved in the development of the model, with the validation of the model, followed by the size effect study with the conclusions at the end of the presentation. Magnetoreological materials are a group of smart materials that are composed of the magnetic particles, either in a polymer matrix or on a carrier fluid, whose properties can be altered upon
Can I interrupt for a second, Dr. Zhou?
magnetic field.
Dr. Cohn pointed out that the screen is very tiny. I agree. Is there any chance, Maharthaj, that you would be able to just share your presentation live?
Yeah, I can.
Okay. Okay. Well, I suppose we'll have to figure out.
Yeah, we cannot from my device because the original video is so tiny. I cannot. Oh no.
Oh, maybe not.
I cannot enlarge it.
Okay. Yeah. This might be a problem. I personally can't read the slides. I don't know if anyone else is having that.
Is it still
It's still very difficult to hear you.
Oh, this is
Oh.
better.
Yeah, this is okay.
Better.
This is better. Is this better for everyone else as well?
Station.
Is it okay now?
Yeah, this should be good. Okay, so then we can start again maybe.
Okay.
On now is the modeling of magnetoreological gels, a study on the particle size effect. So the outline of my presentation, I'm just will be starting off with the introduction, followed by the mathematical preliminaries involved in development of the model, followed by the model development statement and the total stress derivation, surface energy concepts involved in the development of the model, with the validation of the model, followed by the size effect study with the conclusions at the end of the presentation. Magnetoreological materials are a group of smart materials that are composed of the magnetic particles, either in a polymer matrix or on a carrier fluid, whose properties can be altered upon the application of the magnetic field, just as the picture which I have shown over here. The applications that depends upon the basic idea of the magnetoreological materials are engine mounts isolated for the building structures, adaptive tuned vibration isolators, and vehicle transmission systems. So, in general, various experimental works have been already, there has been a vast extensive research on the experimental works on the magnetoreological materials to see how the properties of the materials vary under the influence of the magnetic field. To support the experimental part, a wide research area that is being worked on is the constitutive frameworks on the developed on the magnetoreological materials. Very few works concentrated on developing the constitutive frameworks for the MR gels, but they did not account for the time-dependent behavior of the magnetoreological gels. For the stiffer magnetomaterials, model has been developed to realize this time-dependent behavior. But the continuum-based models have a limitation in their capacity that cannot predict the dependence of the microstructural dependent arrangement of the particles. Micromechanical based frameworks gain more attention since they incorporate the micromechanical details of the magnetic particles, that how they are placed within the matrix. In the mid 90s, Jolly Ethel developed a 1D lattice model to study the phenomena that involved in the magnetoreological gels. Followed by this, Ivanko and his co-workers did an extensive research on the lattice model and successfully predicted the behavior of the MR under the magnetic field applied. The gap which we find over here is, though the lattice model possesses many valuable features, it does not capture the viscoelastic behavior. Also, the study on the particle size effect on the performance of the MR gels and the constitutive model to support the to predict the size effects of properties of the MR gels are very less. So in this present work, We'll be starting off with the microstructure based model will be developed to study the particle size effect, and the surface interaction between the matrix and the particles will be introduced. A time dependent viscoelastic model which has already been developed will be used as a base model. The combined framework will be calibrated and validated using a set of experimental data. This calibrated model will be further used to analyze the particle size effect on the behavior of the emergence. So the present work will be starting off with the mathematical preliminaries, which involves the kinematics of the magnetological jets. Generally, the deformation of the body can be easily tracked down by specifying a point in a material configuration, and will be relocated to the spatial configuration, and this one can be mapped easily using a deformation map chi. The deformation gradient f can be obtained through this deformation map chi and is defined in equation 1. Based upon this deformation gradient, the left and right Cauchy-Green tensors can be given using the equation 2. In general, the deformation gradient is will be decomposed into elastic and the viscous contributions in order to model the time-dependent response of the polymeric materials. Also, since we are dealing with the magnetorheological materials, the magnetomechanics concept also plays a vital role in the development of the model. The fundamental variables in the magnetomechanics are magnetic field, magnetization, magnetic induction, and the relationship between these parameters between the material and the spatial configuration are defined in equation 4. Followed by the kinematics, the balance laws has to be established for the development of the model. The balance laws relevant to the magnetomechanics are defined in equation 5, which are the result of the Maxwell equations. And the equation 5 will be satisfied only when the equation 6 possesses well. The connection between the three parameters, magnetic field, magnetization, and the magnetization and magnetic induction, are shown in the equation 7. The total first pair of Kirchhoff's stress tensor P dot are related using the total second pair of Kirchhoff stress and total Cauchy stress, which are given in the equation 8. On the whole, the total pair stress is given by, or we can consider it is conceived as the sum of the mechanical component, magnetic component, and the Maxwell component. The constitutive equations which are involved in the development of the model are: the strain energy functions of the material can be generally described as a function of the deformation gradient, internal variable, and the magnetic variable, and is applicable for all isothermal case, and it is dependent on the three independent parameters. The total energy is expressed as shown in equation 10, with N0 being the free field magnetic energy. For an isothermal case, the Clausius-Duhem inequality can be expressed as del naught, and which is expanded and expressed as shown in equation 12. Using these, the constitutive relations are defined in the terms of the total energy as shown in equation 30. So we'll be starting off with the model. In general, MRGL consists of magnetic particles embedded in a polymer matrix. The particles, upon application of the magnetic field, respond. In here, the stiffness of the gel is highly influenced by the magnetic particles, since the particles are much more stiffer than the matrix. Various works have shown the enhancement or the improvement in the modulus in both isotropic and anisotropic gels upon the application of the magnetic field. The strain energy per unit volume of the gel composed of the magnetic and mechanical, and the term which we are introducing over here is the interfacial interactions that occur between the matrix and the particles. First, I'll discuss with the magnetic and the mechanical contributions. Later, I'll discuss the interfacial interactions. So, the strain energy can be described as sum of the magnetic contribution and mechanical contribution. So, mechanical contribution is further decomposed into two. which will be realized as a sum of the purely elastic one and viscoelastic component. So the magnetic strain energy, the strain energy functions which will start off deriving. The magnetic strain energy per unit volume is described as shown in the equation 16, where mu naught and mu r are the relative permeability of the vacuum and the medium respectively, where v naught being the volume of the total volume of the MR gel sample. The Mi and Mg term are the dipole moments, and where small vp denotes the volume of the particle. Here, Rig denotes the vectorial distance in the spatial configuration between the ith and the jth particle. Using these information, equation 16 is rewritten as equation 17. Further, bringing in various information like Ra being the distance, capital R being the distance between the particles in the material configuration, it is rewritten to bring in the relationship of the distance between the particles in the spatial and the material configuration as small r is equal to Fri. The reduced magnetization term is used to simplifies the term equation 17. So m bar is used is the ratio of magnetization and the saturation magnetization. Using these identities, equation 17 is further reduced, and where the beta denotes the number of particles per chosen representative lattice, while n represents the total number of particles within the gel sample. Now, the magnetic part is done. We'll move on with the mechanical part. The elastic and viscous contribution of the MR gel associates with the volume fraction. So, the volume fraction of the magnetic particle is considered to be 5. So, the rest of the part contributes to be 1 minus 5. Using Aradaboy's formulated 8-chain approach, the elastic and viscous contributions, which is also a microstructural based one, the strain energy functions are defined using the Aradaboy's 8-chain approach as shown in equation 19 and 20. So in the equation 19 and 20, G and GV represent the shear modulus of the elastic and viscous response of the MR gel sample. And Ne and Nv represent the number of Kunz segments per polymer chain of the elastic and viscous network. Upon 5 summation terms have been used, and since the influence of the particles are higher, I1 has been altered to I1H, which is written as y of I1 minus 3 plus 3, where I1 is the trace of the left Cauchy green tensor. Followed by the mechanical part, the total stress derivation using the magnetic and mechanical part will be defined over here. Using the above mentioned equations, the total stress is given by the summation of the terms shown in 21 equation. So in the equation, it is composed of the elastic part, mechanical part, and the magnetic part. In addition to the components, the surface energy concepts will also be included. I'll show in the which I'll discuss in the later part. The surface stress that accounts for the surface interaction between the magnet matrix and the particles will be introduced. The stresses that are pertaining to the elastic and viscous parts of the mechanical component using the 8-chain model as described above previously will are shown in equation 22 and 23. Similarly, the magnetic part of the total stress obtained using the equation 18 is expressed as follows in equation 24. So now we'll go into the surface energy concepts. The stress that arises due to the interaction between the magnetic particles and the polymer matrix is denoted by the P sub. Generally, the surface area and the mass of the spherical particle whose diameter is d is denoted by Sd and Md and is given as shown in equation 25. And the single particle surface energy is expressed easily in equation 26, where gamma denotes the interfacial tension. The surface energy per unit volume is rewritten for n number of particles as shown in equation 28. Further, using the above equation, The stress due to the surface interaction between the particles and the matrix is deduced and is given by equation 29. This 29th equation will be summed up and the total stress will be rewritten as equation 38. Now the expression related to the stresses will be of elastic stress, magnetic stress, viscous component and the surface stress. All these will be derived, and when put together the total stress can be computed using the equation 30, which will be further implemented in the study of the size effect and how it affects the properties of the emergence. This model is validated by uh, by setting it, by validating it with a set of data which obtained from the previous work. This experimental data is conducted by performing the shear test and isotropic emergence using a free decay test apparatus and at different magnetic field values to obtain its properties. And isotropic gels are made up of carbonyl ion particles with an average size of 3.2 micrometer. The interfacial tension between the value of the interfacial tension of the emergent sample is found out to be 203.55 millijoules per meter square. Further,
Sorry, we still have to jump into the conclusion part for the time limitation. Let's see the
relatable agreement between the experimental and the numerical simulation. So the conclusions which we obtained from the work is that the microstructural based model that captures the time dependent viscoelastic behavior has been adopted. This adopted model is extended to account for the surface interactions between the matrix and the magnetic particles to study the particle size effect on the properties of the emergence. This developed model is calibrated and validated using a previously developed, found out set of experimental data. Further, the size effect and the attributes of gel is steady, and the results reveal that as the particle decreases, the shear stress value increases for the given shear strain values. This behavior is constantly observed for all the three reduced magnetic field strengths. So here are the references which I have used to develop this work. Thank you.
Okay, thank you very much. Does anyone have any questions for Mehardaj? Okay, if not, then let's move on to our next presentation. This one comes from Samuel Mugo at MacEwan University in Canada. Samuel, are you in the Zoom? Doesn't look like he is. I believe he sent in a video. So let's go ahead and get that up if we can. Dr. Zhao, if we could get that video up from Samuel
okay.
Mugo.
Okay.
Yeah.
For the next 15 minutes or so, I'm gonna discuss what we are doing in my research group, which is in MacEwan University. My name is Samuel Mugo, and today I'm gonna talk to you about our work on fabricating microneedles, where we print multi-responsive hydrogel materials, and then we use those fabricated platforms for chemical analytics. Now, whether we are dealing with animals or plants, all of us, tend to respond to stress in very similar ways, possibly due to evolutionary convergence. And plants and animals, produce different types of metabolites as a result of be it biotic or abiotic stresses. And these metabolites are what our interests are in terms of fabricating sensors that can be used to detect these metabolites that can be used as indicators of stress, be it for plants as well as animals. And some of the metabolites that we are interested in include things like hydrogen peroxide, which is the most stable reactive oxygen species out there. Also interested in different types of electrolytes. I'm interested in adrenaline, cholesterol, and so on. But today my focus is going to be mainly on dissolved oxygen. I also going to talk a little bit about hydrogen peroxide. Also, maybe I'm gonna give an example of cortisol, which is probably the most predominant of the stress hormones out there. And I'm gonna talk about pH and redox potential, two of which I consider to be the most important types of chemical equilibria out there that can be used to be bulk indicators of stress metabolites. Now, the analytical challenge out there is, we want to get away from these very expensive types of chemical analytical platforms that are localized in the labs, things like gas chromatography, things like spectroscopies and so on, when they are very, very robust and so on. Unfortunately, they're very expensive and you cannot take them to the field. Now, the future of chemical analysis is where we democratize it to fabricate small platforms that are frugal, that can analyze the chemical species very rapidly. And also very important, that do not disturb the specimen that is being studied. And so you can do what we call the in situ non-invasive type of analysis, and as such you can take these types of platforms in the field, and hopefully you can also hyphenate them to cellular platforms so that you can do data analysis that way. What we are referring to as the Internet of Things, really, which is the future of chemical analysis, as far as helping consumers make decisions from the data that is generated from these chemical platforms. And so sort of, this is the future where we are getting these platforms that are embedded with all these types of features. And indeed, that is our goal, to create mainly electrochemical platforms, that embody all these types of features essentially so that we can democratize chemical analysis and get these platforms out in the field instead of bringing the sample into the lab. Now, very quickly, the different types of architectural platforms that we fabricate includes the planar types of sensors, but you also fabricate the penetrative, types of sensors, which comprises microneedles. That's very important if at all we want to very non-invasively penetrate, be it plants or even animals, so that we can sample biological specimen that may not be on the surface. For humans, there could be things like the interstitial fluid, and for plants, there could be things like the apoplastic fluids, that are just below or inside the bark of the plant. And so we normally use the PDMS to fabricate these things we are calling the flexible platforms. They can be non-penetrative, meaning we don't have microneedles, but also we can fabricate the PDMS, which is a flexible platform, and integrate microneedles, which looks something like this. And we usually do that using very inexpensive platforms created using actually a beeswax mold. But again, I don't have a lot of time to talk about that. You're going to see in the literature the way they fabricate these microneedles is very time consuming. But my group has, again, developed a very inexpensive and simple way of fabricating PDMS microneedles using a mold such as a beeswax. Other than the flexible sensors, we also use the rigid types of microneedles, again, very simply using the hypodermic needles. And then we coat inside, with the molecular receptors that can be used to detect the analytes of interest. And lastly, We also use a textile-based types of flexible materials, and this is essentially just cotton. And then we modify it using different types of conductive materials that I'm gonna talk about later. So very quickly, well, you know, for a sensor to be useful, it needs a selective biomimetic, you know, receptor, which is what we normally ourselves are interested in, things that are receptors or molecular receptors that are very, very stable environmentally. And so again, it makes it easy, you know, to take these types of platforms in the field. And so despite the fact that in my past, we've done a lot of work with enzymes, we've done a lot of work with aptamers. Again, all these are very selective types of molecular receptors, but unfortunately enzymes, again, they are very limiting because they are environmentally unstable, similar to the antibodies. In a way, the aptamers are fairly stable, but again, you still need to refrigerate them. Again, we've done quite some bit of work on that. But my interest and in this presentation is to show you that you can actually use very, very stable types of polymers, what we refer to as responsive polymer hydrogels. And these are fairly well-known materials that change their shape and conformation if you expose them to different environmental stimuli. But also, if at all you want to create even more versatile and yet robust type of platforms, you can create what they call the hybrid metal-organic frameworks, where you can create a hybrid system, probably comprising of the responsive polymers, and then you integrate, you know, things like the metal centers that are fairly useful, especially if at all you're doing electrochemical platforms. And I'm going to show you that you can actually create the metal-organic frameworks also by making them a bit more intelligent using the molecular imprinting method, so that they can be very, very selective to whatever you're interested in. Now, maybe the very first example that I'm going to show you is a planar microneedle platform based on PDMS that we developed to detect pH as well as cortisol in human sweat, and we are doing that simultaneously. And very, very simply, you know, we fabricate these microneedle platforms. I already said that we use a beeswax to create the PDMS microneedles, and then we print it with the carbon nanotubes so that we can have a conductive platform. And then we integrate the polyaniline, which we just do simple radical polymerization. And it's fairly well known, you know, that polyaniline essentially is pH responsive because it interconverts between the meradine type of polyaniline and these other oxidized form type of polyaniline if you expose it to hydronium ions. And so essentially, you know, we tested it, you know, to see if we can use it as a pH sensor. On the other hand, using the very same type of platform, and again, we are printing it, you know, next to each other, so in parallel. You know, we use exactly the same base, microneedle PDMS, printed it with carbon nanotubes and cellulose, which is cellulose nanocrystals. Again, we are using that mainly as a binder, you know, so that the carbon nanotubes can print as a very, very thin film. And then we integrate again the polyaniline, as you're gonna see, as a conductive polymer, so it increases, you know, the conductivity of the entire platform, despite the fact that, of course, you know, you're also using it as a pH chamber. And we integrate on top of it, you know, the polymethacrylate cortisol molecularly imprinted polymer. And again, I've published this work, so you can check, you know, the details there, which is you take the glycidyl methacrylate cross-linked with ethylene diamine dimethacrylate, and using radical polymerization, you know, you create those cavities, and then you extrude or extract the template, you know, which is cortisol in this case, and we do so using electrochemical cleaning method.
So, sorry, this time still have to jump to the final because there are only 10 minutes for one speaker.
Doesn't break at all. Electrochemical sense. So I'm gonna leave it at that. chemical sensors, but also, you can use them to verify what you've analyzed using electrochemistry, by chromatography. And again, because of time, I'm gonna leave it at that, by acknowledging the people who've done the work, my students. Most of the work that I've talked about was done by Wei-Ha Lu, who's a research associate in my group, and also Marika Wood as well. In fact, Marika Wood is presenting, so you can look at her presentation.
Sorry for everyone. I have to stop here because it's already over a little. Sorry.
Thank you, Dr. Zhao. Next up, we have Jiangtao Cheng from Virginia Tech. And I see that he is here. Thank you, Dr. Cheng. Go ahead.
Hello, everyone. So it's my great pleasure to share with you my research on Leidenfrost evaluation assisted ultra-sensitive surface enhanced Raman spectroscopy. Now, SIRS actually has been widely used for fingerprint detection of the metals because the enhanced local electromagnetic field can lead to the enhanced fingerprint signals from the oscillating dipoles in the molecules. And in general, people use the metal nanoparticles, and the gap, with the distance in the order of the tens of nanometers can lead to the strengthened local electromagnetic field. But however, the wide applications of the SIRS technology still needs to overcome several challenges. For example, how to form a locally enhanced electromagnetic field, and how to form and control the distribution of the SIRS active hotspots. And most importantly, how to precisely deliver the analyte of the samples just on top of the hotspots. And also, in general, to achieve high efficiency SIRS signals, we need to concentrate the initially dilute analyte molecules on a very small footprint. So this is a lot of challenges needed to solve before the real applications of the SIRS technology. And in this work, so we used the biomimetic lotus leaf-like surfaces as the SIRS platform. So we can see lotus leaf, there exists two scaled hierarchical structures. So the cilia, that's the nano structures actually grown on the papilla, and the papilla actually is the micron scale small hills. And we used the deep reactive ion etching to etch the micron scale pillars. So and then we grow the carbon nanotubes on top of the micro pillars. So that formed the hierarchical structures actually mimic the lotus leaf-like surfaces. And these surfaces actually forms a platform of the SIRS. So this is the detailed structure of the SIRS platform. So first we etch micron scale pillars on the silicon wafer, and then we grow carbon nanotubes to form the hierarchical roughness. And then we use the electron beam evaporation to alternately form the metal layers. So that's the gold layer, and subsequently followed by the silver metal layer. And in this way, we can form not only the lateral distributed hotspots between the nanotubes, we also form the vertically hotspots along the multi-layer nano antenna, and also the near field FDTD simulation shows that the nano antennas can give rise to the enhanced electric field intensity up to 3 orders of magnitude, and also it can lead to the inhomogeneous, okay, broadening effects. So which means it can exhibit a broadband optical absorption. Now, droplets sitting on the roughness can stay in two states. One is in the cassette state, so which means droplet only in contact with the tips of the roughness. And also, so with the deposition of the metal layers, so metal itself is hydrophilic. So we can say the droplets can actually fill in the interstitial cavities. So this is the Wanzel state. And by controlling the droplet states on the nano antenna surfaces, so we can control the evaporation enrichment of the content analytes. By natural evaporation, so we can see the final footprint is in order of the millimeters. And also it takes hours to have the full evaporation of even the 4 microliter droplets. So this is too long, okay? And then so we applied the Leidenfrost effect. So Leidenfrost effect, which means if we put droplets on a hot surface, so we can see the formed vapor layer can make the water droplet, okay, hovering over the vapor layer, okay. And this is an example, okay, showing the Leidenfrost effect. And this is the hot surface. So we can see the evaporation of the droplets, okay, on the hot plate, okay, actually lead to the relatively fast evaporation rate. But the vapor layer formed between the droplet base actually can finally make the droplet, okay, move away from the hotspot surface. So we prefer to use the partial Leidenfrost effect, so which means the droplets, okay, lifted up along the perimeters. So which means the perimeter region actually in the cassette state, but in the middle, the droplet got pinned in the middle. So this is called the partial Leidenfrost effect. And this is the example showing the natural evaporation. So we can see after the full evaporation, the droplet is sparsely distributed on the relatively large footprint. But with the heating on the substrate, so we can see the Leidenfrost effect lead to the extremely concentrated footprint in the order of the tens of microns. So this is a significant enriched analytes. And this movie showing the partial Leidenfrost evaporation based enrichment of the droplets. So we can see with the evaporation on the hot surface. So this is shows the continuous shrinking along the perimeter of the droplet. But in the middle of the droplet base, it got pinned. So which means the droplet will stay, okay, on the surface. And finally, the significant enriched analytes will be deposited on a footprint only of tens of micron scale. Okay. So we can see that's the pinned analyte droplets, okay, and the footprint is only tens, okay, microns wide. Okay. So based on the partial Leidenfrost evaporation, we compiled the natural evaporation spectrum of the source. So that's relatively very weak signals. But with the Leidenfrost evaporation, so we can see the significantly distinct and apparent source signals. And we can also use the FDTD simulation. So that's the hotspots along the carbon nanotubes, nano antennas. So that's the vertically distributed hotspots. Okay, and also the silicon carbon nanotube hierarchical based plasmonic structures can absorb almost 100% of the incident light, and the source enhancement factors can reach above the 10 to the power of 7. And as a summary, so we're using the writing properties of the source platform and as well as the plasmonic nano antenna structures, we reached very fast analytic source detection in minutes instead of the hours. And also we can detect the nanomolar analytes, and the source enhancement factor can reach above 10 to the power 7. Okay. And I'd like to thank my colleagues, Professor Wei Zhou, and also our students, Junyu Song and Wei Fengcheng, for this work. And thank you so much. I'd like to take any questions you have.
Thank you, Professor Cheng. Are there any questions?
Okay.
Oh, I'm asking the room if there are any questions.
Yes, please.
Okay. If not, let's move on to the last one.
Thank you.
Thank you. So this is for Mohammed K. Abdel Rahman from the University of Illinois, Chicago. Are you in the Zoom?
Yes, I am.
Okay, could you share your screen, please? And go ahead and get started.
So do you see the presenter view or the presentation?
It looks like we're seeing the presenter view, but if this is fine for everybody, then it should be okay.
Better? Okay, so thank you everybody for sticking around to hear my presentation. I'll be talking about propine hydrogenation over palladium copper alloy surface studied using infrared spectroscopy at ambient pressures. My name is Mohammed Abdel Rahman. This is part of my PhD work at University of Illinois, Chicago. For plastic manufacturing and things like that. Common contaminants involved are other C3 hydrocarbons such as allene, propane, and propine. And these are undesired because they can inhibit the polymerization effect of propylene. And so in order to take the contaminant and get rid of it, the best way to do it is to turn it to the desired product, in this case, propylene. So the purpose of this talk is to focus on converting propine or methylacetylene to propylene using partial hydrogenation. So current catalytic techniques often use palladium or platinum-based metals, and those are active for hydrogenation because there's a low energy barrier in hydrogen activation. They split the hydrogen molecule to atomic hydrogen, which migrates across the surface and reacts with the absorbed hydrocarbon. But the problem with those platinum and palladium type metals is they're very active, and you can hydrogenate all the way to the alkane, to the fully saturated product, which is undesirable. On the other hand, using transition metals like copper, they have a high activation energy for hydrogen activation, and so typically polymerization is a thermodynamic pathway for hydrocarbon reactions. But using this single atom alloy approach, we can take a single atom of the active metal and embed it into the inert surface. We can control the limit of hydrogen activation such that we promote the production of the alkene instead of the alkane or polymers. And so we building on work of McQuay et al., who did they laid the groundwork for this project, and they basically determined all the optimal ratios of copper to palladium and hydrogen to hydrocarbon, as well as the temperature that's most efficient for this reaction. So they found at 383 Kelvin, they get 100% conversion, and they have very high loadings of copper. And so here, this 50-to-1 copper palladium ratio provided the highest selectivity to alkene, which is the yellow bar here. But they did not investigate the kinetics of this reaction, so that's what we set out to do. So using infrared spectroscopy, because it's non-destructive, we can identify the infrared spectrum of each molecule, and focus on specific peaks such as 912 wave number for the CH2 bending mode of propylene, or the 3323 wave number for the CH stretching mode of propine. And so we can monitor these peak intensities in real time and use that to determine the rate kinetics, the rate constant, the kinetic order, and the activation energy for the overall reaction. And so first we start by looking at the hydrogenation over a bare copper surface. So we used the copper 111 surface. And after 12 hours at 383 Kelvin, we found that only 5% of the copper of the propine reacted. And so we can see that here by all these spectra looking almost the same, very small intensity change here in 3323, and very small growth of 912 wave number. And so the selectivity and conversion were calculated based on these equations, where conversion is the amount reacted versus the amount introduced, and selectivity is the amount of the desired product formed divided by the amount of the starting material. But when we contrast this to a 2% single atom alloy, we see that within the same 12-hour period, we have complete reaction of propine. So here the intensity of the white spectrum was propine at 0 hours, and the pink one being at after 12 hours. So after 12 hours, basically we've converted all of our propine into propylene. However, when we look at the conversion and the selectivity, we find that we have 100% conversion, but 50% selectivity. So although we have these significant peaks here, 912, 991, and so on for propylene, it only accounts for 50% of the starting material. And another observation that we made is that after 12 hours, we noticed that the catalyst hasn't been poisoned. If it was poisoned, we would have still seen some propine remaining in the gas phase, in the infrared spectrum. So the reaction went to completion with 50% selectivity to propene. So the next step was to carry this out under different conditions to try to determine the reaction order. And so using these three conditions, here I've labeled the reactant amounts in micromoles as well as millitors. Since these are gas phase reactions, we use partial pressures. And so from here, we can determine the rate law, which comes out to this equation. So approximately 1 for the rate order with respect to hydrogen and negative 0.36 with respect to propine. Compare that to literature values, we find that the value for the single atom alloy is most similar to that of pure palladium and not pure copper. And so we found that to be quite surprising, actually, given the fact that we have palladium as 2% over the whole catalyst surface. And so to go one step further, we varied the temperature and we determined the activation energy of the reaction by using a simple Van Hoff plot. And from here, we again compared to the Mann and Colby work from the 60s, and we find that the activation energy is also the same as a pure palladium catalyst. And so what we've done is that by introducing the single atom alloy, by making this single atom alloy, we've effectively lowered the activation energy for the overall reaction in half. And we're showing the same kinetic, same dependence on pure palladium with only 2%. But we didn't account for the other 50% of the starting material. So since we know half of the propine goes to propene, the other half has to go to something else. And so we turn to our other techniques, such as Auger electron spectroscopy. Here we can see that our surface after the reaction in the yellow and the pink spectra are shown to be all pure carbon, as opposed to prior to the reaction, we had palladium and copper surfaces. And using temperature programmed desorption, where we monitor the mass spectrum as a function of temperature, and we see we have major desorption peaks for the various hydrocarbons and their polymers. So we know we have a carbonaceous surface, and unfortunately, we can't identify what the nature of that surface is, other than it's some hydrocarbon-containing species. And this green dotted line in the TPD spectrum is the temperature at which the palladium atoms will dissolve into the bulk of the crystal. And so we lose our single atom alloy surface at that point. So all of our reactions were carried out in this range, 300 to 400. So the surface is still pretty much a single atom alloy. And of course, taking a picture afterwards, we see that there is a black spot here in the center of the sample. And that's where the carbonaceous surface interacted with the electron beam from the Auger source. So to conclude, we found that we can determine the rate equation for the hydrogenation of propine over a single atom alloy surface, and that It has the same energy, it has the same kinetics as a pure palladium catalyst. So that basically tells us that using 2% of the material, we can get the same kinetics with the advantage of the selectivity of the copper here. And typically in catalysis, we report turnover frequencies or turnover numbers. And we reported here 33.2 turnovers per second at 383 Kelvin. And that's based on palladium being the active site. Whereas some literatures will report a lower number using either the whole surface or copper as the active site, depending on where you determine what the rate determining step is. In our case, since hydrogen activation is basically the rate determining step that occurs on the palladium sites, so that accounts for our high turnover number. And with that, I'd like to thank my advisor, Professor Michael Trineri, and the rest of our group at UIC, especially Arif and Islam and Dr. Paul Melnikov for their assistance to the project, and the NSF for funding, and the organizers for giving me the opportunity to present here. At this time, I'll take any questions. Thank you.
Thank you very much. Are there any questions for Mohammed? Okay, if not, then thank you everyone for your wonderful presentations today. Sorry we had some technical glitches in between. That wraps up this oral session, and the next one will be happening at 6:30 p.m. Pacific time. So if you guys are interested, then I believe you can either log off or you can stay on the Zoom and then rejoin at 6:30. But yeah, thank you very much for your attendance and for your participation. And yeah, that's it.
Thank you. Thank you very much, Akshay, for chairing this session.
Yeah, of course. Do you need me for anything else, or can I log off as well? It's kind of late on the East Coast.
Thank you. I think that's all from today. So
okay, thank you very much.
On this platform, we will be joining tomorrow at 8 o'clock.
Okay.
So thanks to everybody for showing up today.
All right. Thank you very much.
Hello.
Yes, please.
So is all we are done for today?
Yes, we are done for the day. Thank you.
And tomorrow, we begin 8 o'clock in the morning.
Yes.
Okay, see you tomorrow.
See you tomorrow at 7:50. Thank you for your support.
Bye-bye.
You've done a great job. Thank you.
Bye-bye.